Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Revolutionizing Ophthalmology Clinical Trials Through Enhanced Accessibility and Advanced AI-Driven Analysis. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the highest quality clinical evidence for their pharmaceutical, biotech, and medical device partners. Their endpoint data solutions have supported clinical trials over 26,000 times in more than 100 countries. Their global team of science, technology, and operational experts have supported over 60% of all FDA drug approvals since 2019. Now, it's my pleasure to introduce our speakers for today's webinar. And first, I'd like to introduce you to my co-moderator for today, and that's Dr. Todd Rudeau. He is the Executive Vice President and Chief Medical Officer at Clario. He is providing medical and scientific leadership across the organization. Todd has nearly 20 years of clinical cardiology and pharmaceutical research experience with a career predominantly focused on drug safety. And next, I'd like to introduce you to Dr. Amit Fasanji. He's the Vice President of Medical and Scientific Affairs at Clario. And there he is. Hi, Amit. Dr. Fasanji has over 20 years of experience in basic and clinical research, image acquisition, processing, analysis, visualization, and biomedical software engineering across multiple modalities and the therapeutic areas. So that's Amit. And next, I'd like to introduce you to Dr. Kelly Dumais. She is the Director of ECOA Science and Consulting at Clario. There she is. Hi, Kelly. Dr. Dumais is a scientist with over 13 years of experience in behavioral and life science research. Kelly has expertise in the implementation of electronic clinical outcome assessments and the development and validation of patient-reported outcomes to support labeling claims. So thank you very much there. Now it's my pleasure to pass the mic over to Todd. So Todd, there's Todd. Hi, Todd. So now it's my pleasure to pass the mic over to Todd. So Todd, when you're ready, you may begin.
Hey, great. Well, thank you, Sonia, for the very kind introductions. Thanks to all of our attendees, of course, for joining us here today. As Sonia said, my name is Todd Rudeau. Very glad to have the opportunity to host this webinar along with two of our experts today, Dr. Fasanji and Dr. Dumais. You can go to the next slide, please. Drug studies for ophthalmologic diseases are a bit unique in the challenges they present for the drug developers, as well as the challenges they present for patients participating in them. Over the next 45 minutes or so, we're going to take you through some of those challenges, discuss some imaging endpoint modalities, discuss the role for centralized review of imaging. We'll also highlight some really interesting and innovative applications of AI and discuss their role in enhancing data insights, improving data quality, trial efficiency. And lastly, there's some very unique considerations for PRO strategies in patients with visual impairment that Kelly's going to take us through. You can go to the next slide. To set the stage here, first though, I have just a brief overview of ophthalmologic diseases and drug development that I wanted to cover. As you see by the numbers here, we're not talking about rare diseases today. Over 250 million people worldwide are afflicted with visual impairment, with about 60 million of them functionally blind. Amongst the leading causes of that blindness are conditions you've likely heard of before, age-related macular degeneration, retinal vascular disease, so diabetic retinopathy being a big one there, and glaucoma. Thanks to advances in recent years, many of those common conditions are finally seeing some legitimate treatments become available, but there's still much more progress to be made, and that's why we're discussing this today. For age-related macular degeneration and retinal vascular disease, therapies designed to prevent growth of new blood vessels have shown promise. Much of this has been through intraocular injections, though, so injections directly in the eye, which of course aren't ideal, but with the limited other options have actually seen quite a bit of uptake in the market, by physicians and patients. I'll touch on that eye injection concept here for a moment and draw your attention to the bottom right of the screen. So drugs for ophthalmologic conditions can be delivered in a number of different ways. Topical eye drops, so we're all familiar with this, solutions, suspensions, ointments, gels. Besides being sometimes a bit messy and inconvenient, the bigger issue is only about 5% actually gets into the eye, and so concentrations are somewhat limited. Systemic medications are another option, so intravenous drugs, subcutaneous injections, oral medications. The issue here is whether they'll actually reach the target in the eye and reach a high enough concentration, to be effective. Or as I mentioned, intraocular injections, so directly into different regions of the eye. So we don't, for example, treat liver or kidney disease by a direct injection into the organ. Why do we need to do this in the eye? The difference is that the eye is fairly isolated from the systemic circulation, the systemic blood flow, and for good reason. There's a protective mechanism that's in place to reduce the exposure of the eye to toxins and infections. It's analogous to the blood-brain barrier. You've probably heard of this before. It's the reason a lot of times when a drug is injected into the vein, it may not actually reach the brain, and because the mechanisms are in place to protect the brain. That analogy between the blood-brain barrier and the blood-retina barrier, as it's called, extends one step further. One of the approaches in neurologic disease is using a pre-treatment to actually enhance the permeability of the blood-brain barrier. The same can be done in the eye to improve the delivery, from a systemic medication. Lastly, on this slide, I'll just mention there's some unique sort of preclinical and early phase considerations with ophthalmologic drug development, with the different delivery methods that I mentioned. Understanding any systemic effects of a drug becomes important if it's absorbed in high enough concentrations. Thinking about the metabolism of a drug within the eye itself is also challenging and important. At the early stages of development program, even just understanding if it's going to be possible to develop your drug as an oral drug or a subcutaneous drug, as opposed to needing to be an ocular injection. You can go to the next. So visually here, we see on the left the different barriers that I referred to before. Again, the goal is to be a protective mechanism, but they do prevent beneficial medications from reaching the targets as well. The blood-retina barrier I mentioned, that primarily limits absorption from the systemic blood into the back of the eye. And then there's some other barriers that are shown there that prevent drug delivery from the external surface of the eye, from the front of the eye. And on the right, you see, thinking about ocular injections, there's a complexity in terms of the technique for the injection and where that injection is actually targeted. So in orange, you see what's called a retrobulbar injection. That means behind the eye. Purple, you see it going into the vitreous, the main chamber of the eye itself, and many other options as well. On the next slide, and before I turn over to Amit, I just wanted to highlight the types of clinical testing and specialized imaging modalities that are often incorporated in ophthalmologic protocols, study protocols. So lab testing for PK profile is in some sense very similar to other therapy areas, but as I mentioned, the unique element here is that assessing systemic exposure from a drug that's administered locally in the eye is important, and also understanding the PK profile within the eye itself is part of this as well. If a high enough systemic exposure does occur, though, then a broader safety strategy is needed within that protocol to look for any systemic effects or side effects of the drug. On the other hand, if the drug is cleared very quickly from within the eye, then more frequent injections may be needed in order to keep the drug level high enough for the drug to work. The other testing besides the PK pharmacokinetic testing, the other testing you're probably fairly familiar with, visual acuity testing, of course, slit lamp examination, that's that test most of us have had where a light is used to examine the structures in the front of the eye. Moisture testing is used in clinical studies of dry eye and has to be standardized. Pressures in glaucoma studies. There's a technique called gonioscopy, which is measuring the angle between two structures, the iris and the cornea in the eye. Again, all part of the sort of clinical testing that would be occurring. And then there's the imaging piece, and this is what you're going to hear much more about today. Some of those modalities include really just specialized photography, so slit lamp photography in the front of the eye, or fundus photography, which is looking at the retina surface in the back of the eye. There's something you will see a lot of called OCT. OCT, optical coherence tomography, is another way to get a picture of the retina, but instead of just looking at the surface of the retina, it's actually breaking down the individual layers in cross section. So it's analogous to a CAT scan in that sense. You can think of it as a CAT scan of the eye, but the difference is instead of using X-ray, it's using light, laser light. Another technique called OCT angiography or fluorescein angiography. These are ways to look at the blood vessels themselves on the surface of the retina. And then the last one here is called B-scan ultrasound, which is yet another technique for studying the back of the eye. So you'll hear more about this in the coming segment. Amit, I'll invite you to come on camera, and we can go ahead and advance, and I'll hand over to you. Thanks.
Great, thanks, Todd. So Todd actually covered some of this already, but I'm going to go through some of the common modalities that are involved in retinal disease. And so if you look at the graphic here, it's sort of a sagittal view of the structure of your eye. I won't focus on the front, which is the anterior segment, but just focus on the back of the eye, which is the retina. So if you've gone to your, a GP or an ophthalmologist, and they use an ophthalmoscope, which is shown here in the graphic, they're looking at the back of your eye. You effectively get what's called a fundus image, and it's color. You can attach optics to a camera and take the same type of image, and you get to see the macula, which is the center of the back of your retina, the optic nerve, and a number of vessels that are coming out of it. You can modify the wavelength of the light and look at different structures in different ways. And so if you use a blue light laser and emission filters on the green side, on the green spectrum, you can get a fluorescent fundus autofluorescent image, which is shown on the bottom left there. And this is particularly useful for looking at geographic atrophy for dry AMD, which is a version of AMD, and I'll cover that a little bit more later. You can also inject dyes, and Todd sort of went through this as well. You can inject something called fluorescein or indocyanine, which is a different type of dye. And using the same sort of filter set that you have with fundus autofluorescence, you can pick up the signal of the dye. And this dye will go into your arteries first, and then eventually go into your veins. And if you have a disease like diabetic retinopathy, the dye will leak out eventually from your vessels. And so it's really easy to see, and it's an important way to quantify the amount of disease. So you can go to the next slide, Sonia. The other modality, and Todd touched on this as well, is OCT. And this, I apologize, there's supposed to be a small rectangle on the graphic there. But effectively, it's looking through the depth of your retina. And there's a graphic in the middle that shows you the cells that make up the retina. And the most important areas of there are anything below the ONL, the outer nuclear layer, down to your RPE, the retinal pigment epithelial cells there. Those are your photoreceptors, your rods and your cones. And I'll talk a little bit more about this when we get to the section on dry AMD. But these sort of layers of cells actually show up as structural features when you actually shine a laser on it in the infrared range. And so that's what OCT is doing. It's basically doing a number of slices top to bottom, and this box on the right here sort of shows that there's one single line across, but there's typically about 100 or 128 slices that are required, and they form what we call a cube scan. And individual slices are called B-scan. So it's sort of akin to an axial scan on a CT. But because those different layers reflect based off of the infrared, you end up seeing these bands on the OCT, and those is effectively what's being segmented by AI or being read by a grader or an ophthalmologist when they're reviewing different pathologies for these diseases. You can go to the next slide. So what I want to talk about in the next few slides is really the importance of making sure that you have a platform for your clinical trials that can support the different nuances for ophthalmology. And this is just a listing of the various vendors that support the modalities that I just covered. And so there's a lot of them. This isn't even an exhaustive list. Most of them are not proprietary formats. So for fluorescein angiography, which is the FA, or the autofluorescence, or the FAF, or the color fundus, which is a CFP, you're going to get, DICOMs, JPEGs, TIFFs. So those aren't, unique files. But where you have issues is with the OCT. So there are 4 major vendors: Heidelberg, Zeiss, Optiview, and Topcon. By far, Heidelberg and Zeiss make up most of the market. They have proprietary file formats. In fact, the Cirrus scanner actually puts out a file called .dcm, but it's not a DICOM. It's an encoded version of their file. So it's important to have a platform that can read them in natively, because what you don't want to do is have a site try to export them out as a different file and then upload them into your vendor's platform, because that's additional workflow. Those sites may not even have the software to do that conversion. So within Clario's platform, we have the ability to read that. So it's just important that you query your vendor to make sure that they have this sort of conversion built into their system. The other thing that's important here is also that these modalities have restricted field of use. So with fluorescein angiography, autofluorescence, and color fundus, they have what's called a degree in terms of the amount of the field of view extent. And so 30 to 45 degrees is typical for most of these modalities. If you want larger field of view, you have to basically take multiple fields together and sort of stitch them together. There are vendors on the market that can do ultra wide field, like Optus, for example, where you sacrifice resolution, but you still get a larger field of view. So you can go to the next slide. So in terms of a platform, what you really want is an end-to-end system that supports site upload all the way through read and reporting, all self-contained within one system. It's common sense. If you have multiple systems involved with sort of handoffs between those systems, it becomes a nightmare to manage, and it's even more magnified for ophthalmology studies where you've got multiple modalities and different workflows that are involved in a given study. And I actually will go through like an example workflow at the very end of my section of the talk here. From the site perspective, this platform needs to be geofenced, and ideally it's cloud-based. So they're uploading basically logging onto a browser-based system, uploading their scans. The system basically interprets the scans and ideally reads the native files and basically displays them to the end user so they can confirm that, yes, this is the scan that I uploaded for this particular subject. And then you also want the system to have automated edit checks in there to make sure that the subject IDs and the demographics of the subject match, so that you don't have a different subject uploaded for a different subsequent time point for that subject. And what we've done, at least in the Clario side, is actually do an automated quality assessment of these images. So OCT, for example, the vendors will actually determine signal strength of the scan. And so that's part of the metadata. And we can display that out to the site so we know whether it's a good quality scan or a low quality scan. But that doesn't really necessarily tell you whether a reader can grade it or if AI can interpret the scan. And so what we've done is basically within the workflow included a step where the images get shunted to the AI and it automatically reviews for quality and lets the site know whether a rescan is required. The nuance here is that that patient still has to be there when the site does the upload. And for the most part, sites are doing the upload after the patient's already left the site. So for this to work, you would basically have to have the site do the upload, wait a couple of minutes for the return of the quality assessment, and then it would let the site know whether they need to do a rescan. From the sponsor's perspective, you need to be able to view any of these images, hopefully after the QC part, so that it's been appropriately de-identified, and view not only the original images, but also any annotations that either the reader has performed or the AI has actually pre-segmented and loaded onto the image, and to view sort of the on-FOSS maps, which I'll show you a little bit later, which are more quantitative in nature. Ideally, this is done within the platform, so they don't have to download the images and then use a third-party system to view them, because again, these are proprietary files, and that becomes painful and expensive. And then for the graders, it's pretty self-explanatory. No third parties means that they actually have the reading within one system. You also have this level of consistency across the different scans. So if you imagine a system setup where readers would have to log into OptiView's system or the Heidelberg system to view and read the scans, their tools are in different places, their formatting and all of this other assessment requirements are all different for every piece of software. If you have a system that's agnostic to the vendor, all of those tool sets are the same. So there's a level of consistency and efficiency that you gain with having all of that within one system. And then this AI segmentation is part of the workflow within a platform that would enable the reader to actually see the AI-based segmentation on top of the images and then edit them as necessary. From the study perspective, you want a contiguous audit trail. If they're all in one system, if all of the images are in the system and never leave the system, then you have a contiguous audit trail. If you have a third-party involvement in that process, you have to piece together the audit trail, which can be a bit of a nightmare. You also don't have to wait for a third party to fix an issue, right? That will cause massive amounts of delays. And obviously it needs to be Part 11 compliant with electronic signatures and timestamps for anything that's done with any image or any sign off within the system. And the last thing is, because they're all in the one system, it's really easy to get reporting out, right? You can tell exactly when a reader completed their assessment and whether it's within a certain turnaround time, whether the QC team has actually completed their QC assessment. If the site is not responding or has not uploaded a scan for a given time point, the system can be proactive and let them know that there's a missing time point, and make sure that they upload those in time. So I can go to the next slide. So I'm going to cover a couple of case studies, one for diabetic retinopathy and macular edema, and then dry and wet AMD. So with diabetic retinopathy, the typical scoring scale is the diabetic retinopathy severity scale, which is known as DRSS. It's the gold standard, and it uses the 7 fields of the color fundus imaging, and there's a sort of schematic there. One of the issues with this is that we have historically have had issues even with sites trying to do 3-field or even a single field and making sure that they position these correctly. So it's really challenging to make sure that the imaging is done correctly. The other thing that we've sort of incorporated within our platform is the ability to stitch these images together. So if you think about your phone where you can generate these panorama images, it works on the same sort of technology. It uses homography and feature detection to stitch these images together, and you can see that on the right. And that dramatically improves the reader workflow because they're not scrolling through images. They can see the entire field of view at the same time. Typically, this is done with a 2+1, and you can see the criteria on the right in terms of the features that the readers are assessing. It's a 12-point scale, but it's scoring grading from 10 to 90. If the 2 readers disagree, it would go to a 3rd reader, and usually a 2-level difference would trigger that adjudication. We found that this type of scale, while it does have some value, it's not as insightful for therapeutic benefit as other biomarkers, particularly in fluorescein angiographies, which is what I'll cover on the next slide, if you can move to that. Sorry, yeah, there you go. So this is just an example of what can be accomplished using AI with quantitative fluorescein angiography. So typically these are a series of images, so usually 20 to 30 per eye. And as I mentioned before, it's effectively looking at the transition or perfusion of the dye from your arteries down to your veins. And typically on the late phase is where you would see leakage. In the early phase images, you'll be able to see microaneurysms, which are tiny dots that are in the field of view, and areas of ischemia or non-perfusion that are also in the early stage. So what the AI model that we have does is actually look through the entire series and pick the actual image that actually has the appropriate early phase and late phase for the analysis. Once that's done, it actually goes in and basically runs and detects the area of leakage, the number of microaneurysms, and then the areas of ischemia quantitatively. And these can be done relative to structures in the eye. So typically around the fovea, you could put concentric rings of given diameter and determine what those features are quantitatively within those regions or through the entire field of view. We can go to the next slide. And this is just the number of steps that are taken by the AI automatically. So it finds a region of interest, it auto-detects the blood vessels, finds the optic nerve, fovea, it finds the eyelid because it needs to remove that before any of the analysis is done, and then it segments those features out. One of the things I forgot to mention on the other slides, for leakage, we can actually separate that out into perivascular and generalized. So leakage that's immediately around the vessels, or leakage that's just in general around the areas of the eye. We can go to the next slide. One of the important things is the vascular trend. I sort of wanted to point this out because obviously if you're doing perivascular leakage, you need to be able to segment the vessels out really well. And the image on the left, you can see the original, and it's really hard to see the vessels. You could probably adjust the window and level, scale, or brightness contrast, and you can pick them up, but then you end up oversaturating areas, particularly if you have a lot of leakage or issues in the eye. So you can see how well the AI does on the far right. It picks up the sort of smaller vessels that are really hard to pick up on the left image. You can go to the next slide. And applying this to an actual use case for diabetic retinopathy, typically these are categorized by non-proliferative, mild, moderate, severe, or proliferative diabetic retinopathy. And those are reader-based assessments and kind of not necessarily easy to judge, but if you have a quantifiable way to do that, you can actually use percentages to subcategorize it. I think a lot of that's going to evolve over time where you don't need to create these buckets that are artificially have these cutoffs because you've got this quantitative variable that's now contiguous or continuous, and you don't need to bucket things in this particular way. But anyway, these can be reversed into a given bucket. You can go to the next slide. In terms of OCT, we have a number of AI models. So do a lot of our competitors in the market have AI models that can segment the various layers in the eye. They work, to different extents, work well or not, and different pathologies that are present in the eye can affect the accuracy of that. So we do always mitigate any risks with the issues with AI not working with an overread by a reader. There's always that step. The AI by itself won't generate the endpoint. But with the layer segmentation, you can basically easily output things like central subfield thickness, which is the ILM to RPE, basically the top and bottom layers of the eye, of the retina, that can be easily extracted. You can choose any one of the layers to actually generate these on-phos maps, which you can see on the far right here. And those maps are basically color-coded based off of the distance between the two layers that you've selected. So you can then take those two images, co-register them across time points and say, okay, this is the delta between these two maps for this particular patient. So it makes it really easy to see those changes in pathology rather than having to scroll through the B scans on the OCT. And I'll get through some of the integrity and volumetric analysis in a later slide. So you could go to the next slide. And this is just showing you an example of the segmentation on an actual slice. Here you can see the ILM, the ONL, ellipsoidal zone, and the RPE that's segmented. And again, this is doing it across all 120 or so slices that are available in the cube scan. And that cube scan is typically 6 millimeters by 6 millimeters. It's a really small area, but it provides a lot of information. And you can see those bands that are making up those cells that I had shown you in the previous graphic. You can go to the next slide. So this is what the quantitative mapping can do or provide to you. So you can look at specific targeted areas using these different regions of interest. What's shown here is a 1 millimeter and 2 millimeter diameter circle around the fovea, but these maps are again quantitative. So the easy integrity here is looking between the pseudozone and the RPE. The retinal thickness is the ILM to the RPE, and you can see visually what's happening with the different areas of the scan there. And this can be done and replicated for GA lesions, for sub-RPE, and then various amounts of various types of fluid as well. And you can also look at intensities of these different layers, not just the distances between the layers. You go to the next slide. And then we can also segment different features that are not layer based. So fluid, intraretinal fluid, subretinal fluid, sub-RPE fluid, macular holes, and inflammatory disease can all be pre-segmented using AI models. And so again, if you had a reader try to do this manually, it would take forever because you have to do it on every single slice along the cube. And so this dramatically improves the workflow for the reader and increases the consistency as well. Go to the next slide. And this is just a library of our current algorithms for AI. Obviously, depending on the vendors you're using, they'll have their own. We partner with the Cleveland Clinic. The Cleveland Clinic actually does, it creates all of these AI models. They use some of our software to do the annotations, to train these models. And so there's a fairly large group and they're really nimble, and so they can create AI models pretty quickly and validate them. And so that's what you need to look for in terms of a reading center for your clinical trials, to make sure that they have this sort of technology in place so that they can rapidly turn around and validate models. You can go to the next slide. Yeah, so I'm going to quickly go through, actually, Sonia, if you can go through the next couple of slides. I won't go through this too much, but with dry AMD, that's more prevalent than wet AMD or neovascular AMD. And not all dry AMD will convert to wet form. It's usually 10 to 15%, but they have stages. So dry AMD will start with an early or intermediate stage where you'll have these drusen, and then late stage you will develop this geographic atrophy, and you can use different modalities, including OCT, to assess those. You can go to the next slide. And the neovascular version of that, so if it does convert, you end up with fluid in your retina. And again, we have AI models that can auto-segment that fluid, and particularly helpful for CNV, which is choroidal neovascularization, is fluorescein angiography. So that's one of the important modalities to assess whether you have a CNV, particularly for dark dry AMD that might have a conversion. You're usually acquiring fluorescein angiography images to make sure that there's not a CNV present. I can go to the next slide. So the typical method for doing a dry AMD assessment, this is sort of the gold standard, is to use fundus autofluorescence to look at the area of the lesion, and it shows up as a dark area in that scan, and you have a grader that will automatically sort of or manually go in and sort of paint the lesion and get the area measurement. So usually an inclusion criteria or efficacy component is looking at the size of that lesion and how it's progressing over time. And it's usually a 2+1 review with a third reviewer looking at it if there's a discordance between the two primaries. And then there's an example of a safety report that's or eligibility report that's outputted there. You can go to the next slide. One of the issues is that the geographic atrophy is sort of late stage. So by the time you've gotten there, you've not really assessed necessarily whether your drug has therapeutic benefit. So if you're looking at that sort of window of intermediate to late stage AMD progression, there are other biomarkers that can be interrogated. You just need to define what defines progression. So is it to GA? Is it to EZ loss, or is it to hypertransmission, for example? And what I'm showing you in the schematic is these various features that can show up, and this is sort of longitudinal. You'll start with drusen, EZ loss, followed by hyperreflective foci, hypertransmission, and then eventually to iRORA. So what you want to do is make sure that you're targeting the right sort of assessment or biomarkers and sort of follow them through the time points with your assessments. So you can go to the next slide. And what we've sort of focused in on is sort of the ellipsoidal zone attenuation. And so it's sort of highlighted here. This was actually done on a phase 2 study as an exploratory endpoint or a secondary endpoint. For the phase 3 for this same drug, the FDA decided that the attenuation, the ellipsoidal attenuation can be a primary endpoint, and the GA-based FAF assessment was actually down to a secondary endpoint. So that shows you the value of sort of looking at these other biomarkers for assessing whether you've got preservation of your photoreceptors or not. And if you go to the next slide, it sort of shows you an example of like, and the case on the left here is intermediate AMD loss, or intermediate AMD with ellipsoidal zone loss. The image on the right shows a more aggressive sort of loss there, and you can see the pink on the actual scan on the on-FOSS image is where the distance between the RPE and the ellipsoidal zone has gone to 0. Anything that's sort of a blue is in that partial attenuation, which is around 20 microns or so. So this gives you a really quick view of whether, and a quantitative view of how the disease is progressing. If you go to the next slide. And you might ask, well, how does that really correlate to functional changes in vision, right? So the Cleveland Clinic actually took the GATHER data and pulled it, GATHER 1 and 2, and pulled it together and looked at visual acuity with respect to ellipsoidal zone loss. And there's a correlation between subjects that have no loss in visual acuity with less loss in ellipsoidal zone. So, and that's the same across for whether you're looking at partial ellipsoidal zone attenuation or total. And there's also a correlation to growth of the GA lesion as well. So if you took that data set and bucketed it down to those subjects that have very slow growth versus very fast growth in GA, there's a correlation to at baseline whether that subject had a large amount of ellipsoidal zone loss versus a low amount. So those subjects that had a little loss at baseline would have a slower growth in GA, and those that had higher loss in or higher ellipsoidal zone attenuation would have a faster growth rate for the GA. So it's pretty insightful in terms of like the ellipsoidal zone impact on these studies. We'll go to the next slide. And you can also do this with iAurora using AI as well. And you can move on to the next slide. One sort of nuance that you can do, and this is again goes back to your selection of the correct reading center that you use for your study. We get asked unique things about doing analysis with these studies. And so this was a study that had a surgical intervention. So there's an injection of stems or cells into the eye, and they're looking to see whether the injection site covers the lesion. And so what ends up happening is that you get a video, you stitch those videos, the frames of the videos together, and you can sort of make out the sort of bleb of where the injection occurred, and you can sort of make out where the lesion is on the color fundus, but it's not the ideal way to look at the lesion. So what we did was register the on-FOSS OCT to the panorama that was generated with the bleb on it. And then now on the far right corner, you can see the outline of the bleb and then the segmentation of the GA lesion in blue. And now you can easily determine how much of the bleb actually covered the lesion. So again, it's just a nuance with this particular type of assessment. And we can go through really quickly to the next slide. And this just shows you the complexity of the workflow. I'm not going to go through this, but you need your platform to support end-to-end these various time points with different modalities, different assessment paradigms, including safety assessments, efficacy assessments that can have different reader paradigms, either a 2+1 or a single read, and then batched reads as well. And so it can be it can get very complex really quickly. So I'm going to now hand it off to Kelly, who's going to cover some of the enhancements to ECOA for ophthalmic studies.
Thanks, Amit. That was great. Yeah, so you just saw some great case examples of collecting and analyzing scans, and I'll be talking today about how to supplement these types of measures with patient-reported outcomes. So in the past decade or so, we've really seen a shift towards a focus on incorporating the patient voice into drug development programs generally. Instead of relying strictly on objective measures or clinical scales, regulators are really pushing for patient-reported measures to ensure that the therapies are informed by patients' perspectives. Payers are also more likely to make favorable coverage decisions for products and devices that are supported by data collected from patients. So when it comes to ophthalmic diseases, while outcomes such as imaging and visual acuity are undoubtedly important to determine treatment efficacy, these objective measures still fail to capture the patient's experience of living with the disease, right? So for example, their quality of life, disease symptoms, their emotional functioning, so the stress associated with their visual impairments, physical functioning, their ability to do work and activities due to their visual impairments, and even treatment preferences. So the gold standard for collecting these are through the use of patient-reported outcomes. And the most critical part of patient-reported outcomes, or PROs, is that they collect symptoms directly from the patient, so without any interpretation by a clinician or anyone else. And I read an interesting study recently, which was investigating a new PRO for glaucoma treatment, where both patients and clinicians were interviewed, and both groups were asked to separately identify impacts of the disease to patients' functioning and quality of life. And of the impacts identified by patients, 45% of them were not identified by clinicians. So meaning that clinicians were not aware of almost half of the symptoms that patients were reporting were important to them. So this just really highlights the importance of hearing directly from patients, because they really know their symptoms best. If you go to the next slide, here you'll see in general, the 3 different types of PRO measures that have been used in vision-related research. So they're you have your generic PRO measures, and these are used to measure quality of life in both ocular and non-ocular conditions. So it's therapy area agnostic. So you can think of this as your SF-36, your general quality of life questionnaire. Then we have general vision-related PRO measures. So these are used to measure specifically vision-related quality of life and functioning, but they're not specific to any one disease or indication. And then on the right, you have your disease-specific PRO measures. And these are the ones designed to measure disease-specific quality of life and functioning related to vision. For example, like a questionnaire specifically asking about macular disease, quality of life. So which PRO you use will really depend on your study, and not all ophthalmic indications have those disease-specific PROs, especially the more rare ones. And while in some cases it might be important to develop those disease-specific PROs that might be more sensitive to your intervention, the use of the general vision-related PRO measures can still really be valuable. And just noted here, one kind of high impact example of using PROs is in a comparative effectiveness research. So when 2 interventions are being tested that might have similar efficacy via traditional measures like visual acuity that might be comparable, one way to differentiate them could be on how they impact visual functioning, or how there might be differences in treatment preferences for the 2, such as patients preferring perhaps the dosing type or frequency of one over the other. And these are the types of things, this differentiating factors can really be assessed well via the use of PROs. On the next slide, you'll see, and I'm not going to go into this in detail, but these are just the FDA guidances that are available to you that really outline how to incorporate PROs into your drug development program. So there's the pivotal 2009 FDA PRO guidance that will soon be replaced by these 4 patient-focused drug development guidances. Some are still in draft here. And they really just provide information on how the FDA will evaluate the appropriateness of PROs when used to supporting a label claim. So including how to select a PRO measure, if the PRO measure is fit for purpose for your study population. Again, I won't cover them in detail now, but I leave these here for you as a resource, and they can be really helpful when determining, how to select and incorporate PROs into your drug development program. And on the next slide, you'll see, the use of PROs in ophthalmic indications and trials is not new, of course, and there is evidence of their use even to support labeling claims. So this is an analysis that we did on approved drugs in ophthalmic indications which have PROs or other clinical outcome assessments, or COAs, on their labels. So we used eProvide's Pro Label database and found 57 drugs approved with COAs on the label. And provided here is that list of the top indications where COA-based labels are found. And the interesting part here, I think, is that when you see the graph on the right, only 28% of these included pros in the label, where the other studies used other COA types like Clinrose or they were unspecified. So, and also of those specified, the most common pro used was a general vision related quality of life measure, so the National Eye Institute Visual Function Questionnaire. And really, given the regulatory landscape around pros, I would expect to see that this percentage of pros will increase over time. And also that we might be seeing more disease-specific pros will be used, given that we know that the disease-specific symptoms really vary from indication to indication. So, and the regulators are really paying attention to making sure that what we're measuring from patients in these trials are important to them and to their disease. So I think we might see a trend towards the use of more disease-specific measures. But you see here that it's not new that pros are being used, and I think we'll see a trend for them being used even more. On the next slide, so now you'll see, or you've heard, why you should collect pros, what types of pros are out there and available to you, but how do you collect them, right? So for the best data quality, reduce missing data, reduce inconsistent data, electronic collection or ePro, electronic patient-reported outcomes, are really the best approach. And so pros can be collected on tablet devices, handheld devices, on the web, on the patient's own device. Our science team can help with strategies on not only what pros to collect, but also the best devices to use and the best eCOA approach, and whether that's at site collection or at home collection. But another advantage that I want to highlight next is that the advantage of electronic solutions is that they can also enhance our ability to provide more accessible options to patients, right? So on the next slide, you'll see, currently there are kind of limited or suboptimal options for those with various levels of visual impairment to complete patient-reported outcomes. And perhaps instead of even a true pro that's used in your study, maybe you're using an interview-administered assessment, which might be used when a patient can't see well enough to read the assessment on their own. But this has risk of adding some bias if it's not done correctly. And so I think we as technology providers kind of have a role and responsibility to do our part to make clinical trials more accessible by leveraging this technology to make it easier with those with visual impairment to complete ePros on their own when it's possible. So we sought to do this, and we sought to add a zoom in, zoom out feature to our devices. And this is, where you pinch your fingers on the screen to make the text larger. And it's interesting to think about why this hasn't yet been an option for use in clinical trial ePros, the zoom functionality. This is not something new. We use it in our everyday life on our phones all the time, right? Seems like an easy solution. But for clinical trials, the introduction of these types of features has been seen as a risk because we don't know how they might impact data integrity. On the next slide, you'll see, because including accessibility features such as the Zoom can change the way pro questionnaires are presented to patients, right? And if the presentation or mode is changing, industry best practices tells us that we need to validate that new presentation. We can simply turn these features on, but without validating them first, we don't know how what risk there is to data integrity. So what we did before turning this feature on was conducted a research study to assess the measurement equivalence of ePros delivered with and without the Zoom functionality. So on the next slide, you see the methodology that we used here, and I'll go through this quickly, but in order to show that the Zoom feature doesn't change the way participants respond to questionnaires, we need to show that their answers are the same, whether they completed the questionnaire with Zoom or without Zoom. So we had participants complete the same questionnaire twice, once with Zoom and once without Zoom. And if they give the same answers, it shows that the data is comparable, that implementing that Zoom feature didn't change the way they're responding. The questionnaire we used included all the common response scale types, so it was a general quality of life questionnaire. We had 53 participants, so they came in, and if they were in group A, they would first complete the questionnaire as normal, so without Zoom. They would do a distraction task as a washout, and then they would complete the same questionnaire, but this time with it all the way zoomed in. And then we counterbalance this so that the other group first had it zoomed in, did the distraction, and then had it without the zoom. And if the Zoom made it so scrolling was required, they were only able to scroll vertically and not horizontally. So we were ensuring that they were seeing everything on the screen before going to the next screen. And then after the questionnaires, we did a subset of participants were involved in a usability testing interview. So on the next slide, you see a little bit of demographics. This is just our participant demographics for the study. We did want to make sure that our study findings were generalizable to the wider population, but note we did recruit participants that had visual impairment, so almost 70% of participants wore corrective measures, and we also had a good age range there between 28, aged between 28 and 80 with an average age of 53. So if you go to the next slide, this shows our results, and we were very happy that the data showed that it was equivalent. So remember, we wanted to test whether the zoom in feature itself would change the way participants respond. So to determine that, we wanted to see correlation between the answers they gave when it was zoomed in and zoomed out. So a high correlation would show similarity in responses, and we did see a really high correlation here. All response scale types was above 0.9 correlation. The 95% confidence interval was above 0.8. So these are really strong findings, and thereby concluding that the zoom in out feature does not itself change the way patients are responding to questions, and so data integrity is maintained. I think it's important to remember that, though, whenever you're implementing eCOA, it's important to follow implementation best practices, ensuring that participants see everything on the screen before proceeding. Not all Zoom is the same. So when implementing Zoom on your study, you do want to consider certain safeguards, like making sure parameters are in place to make sure that patients are seeing everything on the screen before moving forward, and that kind of aligns with what's been scientifically tested. And then really quickly, the usability on the next slide, we were very happy to see that the functionality was very easy to use by participants. So we asked how easy or difficult the feature was to use. You can see they found it very easy to use, saying things like, I didn't have to think about it. It was very intuitive. We also asked them how useful they thought it would be, also considering their visual impairment. They said it was very, they would think it was very useful. They said things like definitely useful. They found it easier to read the text. So this is very positive feedback that we were really happy to receive. And then my final slide next, we're wanting to make sure that we're providing lots of features, making our devices more accessible to a wider range of populations, right? So this is another feature that we're really excited about that we're launching now. So it is available now, and it's a dark mode feature. So participants can toggle on the dark mode setting, which will have, lower light emittance. So in addition to just adding another layer of patient preference, a lot of, patients might just prefer this dark mode. It can really help those with light sensitivity. So migraine is an obvious use case, but it can also be very helpful, I think, in some ophthalmic indications where light sensitivity can be either a symptom of the disease, such as dry eye syndrome or cataracts or glaucoma, or maybe even in studies involving eye procedures or surgeries where we know light sensitivity can be a temporary response to those procedures. So we're really excited about this one and the zoom in and zoom out feature and what it means for increasing accessibility. So I hope this was helpful in providing some evidence of the importance of incorporating pros and ophthalmic indications and some best practices for how to implement them electronically. So I'll turn it over to Todd now.
Super. All right. Well, a sincere thank you to both Amit and Kelly for their presentations today. As promised, quite a packed session here, dense with really important considerations for ophthalmologic clinical studies, both from the standpoint of imaging modalities and AI-enabled analysis techniques, but also from the patient perspective and how we can best capture patient-reported outcomes. I spoke to the unique challenges for drug development from the delivery methods and managing the ocular barriers, challenges associated with visual impairment and the management of patients within the study specific to this. Amit nicely reviewed different modalities with us, when their use may most be appropriate. The role for AI here is particularly exciting, enhancing detection of drug efficacy, for example, in dry AMD, leveraging AI to improve precision of data in grading severity of retinopathy or macular edema. And again, the pivotal role for electronic outcomes assessments and specifically patient-reported outcomes, including the role for implementing adaptive technologies that importantly have been well validated to enhance the accessibility of the study without any adverse impact to quality. So with that summary, we're going to transition to a very brief Q&A segment here now. Please continue to use the chat box as you have been to enter your questions. We've had a good number come in. Actually, we're not going to get to all of them, and we will commit though to follow up directly with any of you who have asked a question that we don't get to today, and then just follow up offline. So I see Amit and Kelly have joined. I'm gonna, I think, start with Amit in this one here, if I could. Is the use of AI in ophthalmology studies accepted by regulatory agencies? Are there any strategies that should be in place when AI is utilized? So probably a common question here. Go ahead.
Yeah, thanks, Todd. Yeah, that's a good question. So regulatory agencies haven't specifically endorsed any AI model or algorithm for use in clinical trials. It's going to be kind of hard to because a lot of different groups are creating these AI models. And so what I showed you in the previous slides in terms of what we've done in easy attenuation for dry AMD, the FDA was aware that AI was being used in those studies and is part of the workflow. But what we do when we incorporate AI models, particularly for endpoint generation, is make sure that we have specific guardrails in place. And one of the important things is first is make sure that the AI is not outputting the endpoint on its own. There's some type of overread of that process because AI is not perfect. You're going to have pathology in specific subjects that can cause an issue with the AI, and you need somebody that's experienced to actually edit and adjust it before the endpoint's actually finalized. And so we typically use a sort of sequential workflow where a junior grader will review it, make some edits, and the senior grader will review it and then make some edits before it's finalized. You could do a 2+1. That gets kind of tricky in terms of defining cutoffs for triggering the adjudication. The other thing you want to make sure is you version your AI model and you make sure it's locked down for the entirety of the study. Don't try to retrain the model while the study's in play because you're going to effectively adjust the endpoint like in terms of consistency. So lock it down. If you do need to implement a change to the model and have to absolutely run it in the trial, make sure you run it across all the data. You're not just doing it on future time points.
Great. Yeah, I think that last point's really key from a data quality standpoint. Next one here is for Kelly. Pretty straightforward. When using patient-reported outcomes in ophthalmic indications, where in the endpoint hierarchy are PROs typically placed?
Yeah, good question. So where exactly the pro will be placed will depend a bit on the study's objectives, obviously, and where the different concepts of interest fit within your endpoint model. Some indications also have greater impacts to quality of life or to visual functioning than others, so that will dictate a bit about where pros may fit. Generally speaking, with objective measures and imaging endpoints and visual acuity supporting more of your primary endpoints, the pros are typically more commonly placed within your secondary or exploratory endpoints. In the analysis I showed with the POA-based label claims, those were, most pros there were secondary, with some being exploratory, but most were falling within the secondary endpoints, but it will really depend on the study.
Okay, great. Well, again, apologies to those of you who have asked questions that we didn't get to, and we do commit to follow up directly with you. Sonia, I'm going to turn back over to you now to close us up.
Okay, well, thank you very much, Todd, for being my co-moderator. Thank you, Amit and Kelly, for answering those questions. We have reached the end of the Q&A portion of this webinar. And for those of you interested in hearing more from Clario, American Society of Retina Specialists, on July 17th to 20th, the team will be there in Stockholm. And if you'd like to connect with Amit to schedule a meeting, I'm going to put his email address in the chat box for everyone to grab. And there it is. Okay, and then on July the 28th to August the 1st, the Clario team will be attending the Alzheimer Association International Conference. And here you go with a link to register for that one. And I'm going to put that in the chat box now. And there you go. And then save the date, as the team will be back here at X-Talks on September the 17th, advancing MS clinical trials, integrating medical imaging, precision motion analysis, and ePro data for enhanced outcomes is the title. So please just save the date there. Look for them on September the 17th. And remember, if we did not answer your question, or if the team did not answer your questions, just like Todd indicated earlier, the team at Clario will follow up with you after this presentation. And if you have any further questions, please use the email address that you see there on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event, and a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. And additionally, I'm going to share a link with you in the chat box momentarily, so give me a few seconds to get that, and you'll be able to view the recording of today's event with that link, and also share that link with your colleagues once they register for the recording as well. So I'm going to do that momentarily, as I said. Now, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. I'd like to just thank Todd again, Amit, and Kelly for their presentation and spending their time here with us today. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care, and bye for now. Bye, everyone. Bye, Todd. Bye, Amit. Bye, Kelly. Bye, everyone.
Thanks.